Hello, everyone. I will present you work in collaboration with the Universities of Crete and Warwick on how AlphaFold models can inform the prediction of functional LIR-motifs. LIR-motifs are short linear motifs related to autophagy, a catabolic process conserved throughout eukaryotic species under stress conditions. Autophagy helps cells recycle cytoplasmic material for producing first building blocks to be used by the cell. As such, autophagy is key in several physiological processes, and its deregulation is associated with several pathological conditions. There are different types of autophagy, all of which lead material to be degraded to lysosomes or the vacuole. Central to this talk is macroautophagy, which we will simply call autophagy. This is a process initiated by formation of a double membrane cyst, the phagophore, thought to emerge from the ER, which gradually elongates and finally encloses cytoplasmic material, forming an autophagosome. Autophagosomes might fuse with endosomes and eventually lysosomes, where degradation occurs and release of metabolites. Importantly, the phagophore and autophagosomes membranes are decorated both in the inner and outer side with the members of the ATG protein family, small ubiquitin-like proteins which are covalently linked to membrane lipids. Initially, autophagy was thought to be a bulk process where under different types of stress, cells indiscriminately lead cytoplasmic material to degradation. However, during the last 2 decades, accumulating experimental evidence has highlighted selective forms of autophagy where a wide range of different cargoes can be selectively targeted to the autophagic machinery. Key to this selectivity are the membrane-anchored ATG8 molecules, which can provide docking sites to selective autophagy receptors who bring cargoes to autophagosomes. What is frequent among these interactions is that cargo receptors carry a short peptide with two conserved aromatic and hydrophobic residues regularly spaced in the sequence. These are able to dock to two distinct yet very well conserved hydrophobic pockets on the surface of ATG8 proteins. Several of these so-called LIR-motifs have been experimentally characterized in the literature. What is important is that these interactions seem to be very well conserved throughout from unicellular eukaryotes to mammals. Through the years, different definitions of these motifs have appeared in the literature. All refer to a common core motif with a conserved aromatic and hydrophobic residues. Regular expression-based detection of such motifs, even though very sensitive, it can lead to large amounts of false positives due to the degenerate nature of the motif. In general, short linear motifs with binding function have been often reported in the literature to reside in intrinsically disordered regions, either by experimental evidence or through prediction. However, when LIR-motifs are concerned, there is very little hard experimental data on IDRs and LIR-motifs. We confirmed this very recently when curating literature for the autophagy-related protein thematic dataset available in the June 2022 DISPROT release. So a question naturally following this observation is, do functional LIR-motifs preferentially reside in IDRs? Here we can see the predicted three-dimensional structure of human P62, the prototypic selective autophagy receptor. It is evident that the LIR-motif and its flanking regions are predicted by AlphaFold with low confidence. And low confidence in prediction has been recently associated with intrinsically disordered regions. To answer our questions, we rely on high-quality data from the LIR Central database, which we recently released online. LIR Central contains experimentally validated LIR-motifs manually curated from peer-reviewed publications. We concentrate on the human subset of LIR-motifs to avoid any species-specific compositional biases. Within proteins with available AlphaFold models, we could map 171 LIR-motifs. Surprisingly, this dataset was perfectly balanced in terms of positive and negative LIR-motif instances, which are very important as we will see in the following. We also collected AlphaFold models from the AlphaFold database, and we extracted confidence value using in-house Python code. These values were averaged for the core LIR-motif and its up and downstream regions whenever available, as well as for the overall segment. In addition, we developed individual position-specific scoring matrix models, both for the positive and for the negative motifs, taking into account the overall segment or LIR-motif and its flanking regions. A first observation and interesting result is that functional LIR-motifs tend on average to be more disordered compared to non-functional ones. This is true both for the core regions and for the up and downstream regions to the motif, as well as for the overall segments. However, we can still see that some non-negligible overlap in the respectable distribution exists. Moreover, using the individually trained PSSMs to capture information both for positive and negative examples, we can see that the models can discriminate between the two classes fairly well, even though, again, there's significant overlap. However, when we examine the contrast between the two PSSMs, that is the difference of the scores for the positive and negative PSSM for its LIR-motif instance, we can see that the distribution overlap becomes much less, indicating that this kind of information could be suitable for feeding into predictive models. We went on to combine the AlphaFold confidence scores with the PSSM construct scores to feed simple machine learning models with excellent results. Here we show the results for a simple multi-layer perceptron under a 10-fold stratified cross-validation experiments with excellent results. At the moment, we're working on validating methods dedicated for predicting intrinsic disorder and their ability to inform LIR-motif prediction methods. With this, I would like to thank funding agencies and the University of Cyprus for support, our collaborators, but also the ISMB function COSI organizers for the opportunity to present our work. Finally, thank you all for your patience, and I'm happy to take any questions.